Hello everyone, welcome back to our channel. Today we are talking about the hepatitis C virus or commonly known as HCV. We are going to break it down and help you understand what is it and how it affects our body. So let's get started. So what exactly hepatitis is? In simple terms, hepatitis means inflammation of the liver. Think of it like when you get a cut and it swells up, that's inflammation, and it can happen to organs like liver. And today we are focusing on hepatitis C, which is caused by hepatitis C virus. Hepatitis C virus mainly targets the liver cells, and it is pretty good at hiding from our immune system. In about 70% of the cases, it can cause long terms of the chronic infections as well if it's not treated. And about 20% of the people might develop serious liver problems like cirrhosis, and some of these could also get liver cancer. So, hepatitis C can cause three types of infection: acute infection, that is a short term infection. That can last up to 6 months. Sometimes the body can fight off the virus on its own, but more often it turns into a chronic infection. Secondly, we have chronic hepatitis, and this is a long term infection that can lead to a serious health problem if not treated, including the liver damages and cirrhosis. Third, we have fulminant hepatitis. That's very rare, but a very severe form and leads to liver failure as well. So, what is inside hepatitis C virus? Hepatitis C virus is an enveloped RNA virus, and it has an envelope with glycoproteins and a single strand RNA genome. The RNA genome is pretty unstable and prone to mutations. Let's break down the hepatitis C virus genome over here. So, its genome is about 9600 nucleotide bases and has up to 10 genes. 3 genes are structural, that is E1, E2, and C, and the 7 non structural genes starting from NS1 to NS5B. So NS1 helps in viral assembly and release, NS2 act as cysteine protease and a cofactor in viral assembly, NS3 is a bifunctional enzyme with serine protease and helicase activity. It helps the virus to avoid the immune response. Very important one. Then we have NS4A, that is the smallest and non-structural protein with multiple functions. NS4B induces the change in the cytoplasmic membrane and mediates the virus host interactions. NS5A regulates the viral replication and assembly as well. The last one is NS5B, that is an RNA dependent RNA polymerases that is involved in viral RNA synthesis. Now, let's talk about the replication cycle, or you can say the life cycle. So, hepatitis C virus mainly replicates in the liver cells, and sometimes it can replicate in blood cells as well. First of all, a virus enters the host cell through the interaction between viral glycoproteins and host cell molecules like CD81 and LDL receptors. And the virus came inside by the process of endocytosis. And once it's inside an early endosome, the viral envelope fuse with the endosomes, releasing RNA into the cytoplasm, acts as messenger RNA and produce the polyproteins that is processed into the structural and non-structural protein. RNA application also involves NS5B that we have previously discussed, that is RNA dependent RNA polymerases. It helps in producing new viral genomes. Then the viral assembly, the new virons are then released into the secretory pathway of the host cell. So hepatitis C virus spreads mainly through the contact with infected blood. Some common ways are sharing needles, and it can also transmit it from mother to children. Then there are much more activities like tattoos or piercings. Then blood transfusion or sexual contact can also transmit it. You can prevent it by avoid sharing the needles or injected equipments, and also avoiding the contact with infected blood. The good news is that there are direct acting antiviral treatments that can cure more than 95% of the people with hepatitis C virus within 8 to 12 weeks. These treatments significantly reduce the risk of the death and the development of the serious liver problem.